Director of Regulation and Morphology of Bottlenose Dolphins. But she currently works for NOAA as a program administrator for the Marine Mammal Strandings program, which covers the entire Southeast United States, from North Carolina all the way around to Texas, including Puerto Rico and the U.S. Virgin Islands. So it's quite a challenge. It's a huge territory. And today she's going to be talking about dolphin die-offs in 2013. I'd like to welcome Dr. Fougères.
Thank you. Thanks so much for having me here today. I'd like to start off by thanking the literally hundreds of people who have been involved in responding to and investigating the unusual mortality events that I'll be discussing today. And these data and analyses really belong to the people that are listed here, and so I'm happy to be presenting today on their behalf. So in the United States, all marine mammals are protected under the Marine Mammal Protection Act. And the MMPA, we call it the MMPA because we have to have an acronym for everything, as Bill mentioned, consists of 5 parts or titles, but I'll be focusing on Title IV today, which is most relevant to our talk. Title IV was created by the 1992 amendments to the Marine Mammal Protection Act and established the Marine Mammal Health and Stranding Response Program. NOAA's National Marine Fisheries Service was designated as the lead agency to coordinate all related activities for the whales, dolphins, porpoises, seals, and sea lions. So we don't do manatees, sea otters, or polar bears or walruses, but everything else. And these activities include collecting and disseminating data on health and health trends, correlating health trends data with biological, physical, and chemical environmental parameters, and coordinating effective responses to unusual mortality and morbidity events. So there's many components of the Marine Mammal Health and Stranding Response Program. It includes the Stranding Network and its associated rehabilitation and release activities, the Disentanglement Network, unusual mortality event investigations, biomonitoring and health assessments. There's an associated grant program. It does tissue bank and quality assurance, and we also do data management. But the two that are most relevant to today's discussion are the Stranding Network and unusual mortality event investigations. So volunteer marine mammal stranding networks exist throughout all coastal states to respond to marine mammal strandings. And nationwide, there's more than 100 organizations in the Stranding Network, including nonprofits, aquaria, universities, and government agencies, among others. Together, they respond to more than 5,000 marine mammal strandings every year. So what exactly is a stranding? Well, sick, injured, or dead marine mammals found along our shore are considered stranded, as are entrapped or disoriented marine mammals that are unable to return to their natural habitat without assistance. And there's several types of strandings. The single stranding is the most common. There's mom-calf pair strandings, where the mother might become sick or debilitated and the calf will follow her into shore. And there's also mass strandings, such as the 2 recent pilot whale mass stranding events that you might have heard about in the news off Southwest Florida. In the Southeast region, which, as Kendra said, is from North Carolina through Texas, Puerto Rico, and the U.S. Virgin Islands, there's just under 5,000 strandings over the past 6 years. And there's about an average of 823 strandings per year. And as you can see, the majority of those occur in Florida, North Carolina, and Texas. An unusual mortality event is defined under the MMPA as a stranding event that is unexpected, involves a significant die-off of any marine mammal population, and demands immediate response. There are 7 criteria used to determine if a mortality event is unusual, and these include higher than expected numbers of stranded marine mammals, a temporal or spatial or demographic shift in strandings, similar or unusual clinical or pathologic findings, or mortality in a vulnerable or declining population. And typically, we define higher than expected as 2 times the standard deviation above the average stranding rate. So when we start to see an increase in or unusual patterns in strandings, we consult with a federally appointed working group for marine mammal unusual mortality events, which is a really long name. And they're a congressionally mandated panel of marine mammal health experts with expertise in biology, toxicology, pathology, ecology, and epidemiology. The working group's primary role is to determine when an unusual mortality event is occurring and to help direct the response and investigation. So once a UME is declared, on-site and off-site coordinators are designated to liaise with the working group and to help establish the investigative teams. And the investigative teams investigate the causes or contributing factors to the UME, and they also help develop a work plan for review by the working group, which helps to provide funding underneath the UME Contingency Fund. So this is a generic overview of the UME investigative process, which is modified depending on the specific situation. It's an iterative process, ruling in or out potential contributing factors. So first-tier testing typically tests for the most common or apparent causes of UMEs and evaluates the histopathology to look for common causes of mortality. Much of the testing that can be conducted is dictated by the state of decomposition of the animals, and many times the causes of UMEs are unfortunately never determined. From 1991 to the present, there have been 60 formally recognized UMEs in the U.S., and it's fairly evenly split across coasts, with about a third occurring on either the Atlantic, Gulf of Mexico, or Pacific coasts. The causes of most UMEs, as I said, are frequently undetermined. However, for those UMEs where causes can be determined, biotoxins, shown in red, and those are typically like red tide or domoic acid, followed by infectious disease in blue, are the most commonly found causes. Cetaceans, the whales, dolphins, and porpoises, shown in blue, are the species most often impacted by UMEs, followed by pinnipeds and then by manatees. So today I'm going to tell you 3 stories of unusual mortality events that either began or occurred in the Southeast U.S. in 2013. For each, I'll try to set the stage for events leading up to the UME, review the UME itself, and discuss the current state of the investigation. And these UMEs are the Mid-Atlantic Bottlenose Dolphin UME, the Indian River Lagoon Bottlenose Dolphin UME, and the Northern Gulf of Mexico Cetacean UME. We'll start with the Mid-Atlantic. To begin, I'd like to provide a brief overview of the main animal impacted by this UME and all the UMEs I'll be discussing today, the bottlenose dolphin. Bottlenose dolphins range widely in the warm, temperate waters of the Western Atlantic from maritime Canada to Argentina. They exploit diverse habitats and have a relatively plastic social structure. They're long-lived top predators with lifespans of 40 to 45 years for males and more than 50 years for females. And there's 2 distinct ecotypes, the coastal and the offshore, which differ in their physiology, morphology, and ecology. Offshore animals tend to be larger and deeper diving than their coastal counterparts. Along the east coast of the U.S., there is a combination of an offshore stock and coastal transient stocks that move north and south, typically on a seasonal basis, as well as resident stocks in some embayments and estuaries. In total, there's 15 separate stocks that are listed here with their population sizes noted in the parentheses next to them. And for those where we just don't know, there's an unknown. So we have the offshore stock, the 5 coastal stocks, 2 of which are transient, and 3 of which are resident coastal stocks. And then these are our 15 estuarine stocks that are residents. Although the coastal transients or migratory stocks range up into the waters off New York in the summertime, resident animals exist in the warmer waters from North Carolina south through Florida. Bottlenose dolphins are the most common dead stranding in the Southeast U.S., making up 85% of all dead strandings. And this is due to the fact that they live in coastal habitats where they're more likely to wash onto shore when they die, as opposed to pelagic animals that die further out at sea. And they also less commonly live strand unless they are an offshore bottlenose dolphin, likely because they're more familiar with navigating the shallow coastal areas that they inhabit. Bottlenose dolphins in the coastal stocks along the east coast of the U.S. are currently listed as depleted or below their optimum sustainable population level. due to an outbreak of morbillivirus that occurred in 1987. This outbreak was also the impetus for the amendments to the Marine Mammal Protection Act that codified the Marine Mammal Health and Stranding Response Program in 1992. During this event, 742 bottlenose dolphins were reported stranded between June 1987 and March 1988 from New Jersey through Central Florida. Strandings first increased in the waters off of Virginia, over here in June, and then they moved north to New Jersey into the summertime and then south into Florida in the winter. Our understanding of dolphin stock structure at the time was not as comprehensive as our current understanding, and the stranding pattern observed during the outbreak led researchers to theorize that there was a single coastal migratory stock, of which an estimated 50% died during the event. The die-off was initially attributed to brevetoxin due to an unusual red tide off the coast of North Carolina during the same time frame. However, in 1994, retrospective analyses of tissues archived during the event found a virus, unknown at the time of the outbreak, called morbillivirus, in 53% of the dolphins examined, making it the first confirmed outbreak of this virus in U.S. waters. Other outbreaks have since been identified in harbor seals and striped dolphins in Europe and in bottlenose dolphins in the Gulf of Mexico. So what is morbillivirus? They are RNA viruses that are similar to the human measles or canine distemper virus. They're highly infectious and can cause significant illness and death in cetacean species. Morbillivirus spreads via inhalation through respiratory particles or droplets, or direct contact through the eyes, mouth, stomach, skin wounds, or urogenital tract. It can also spread across the placenta from mother to calf. The virus replicates in many different tissues, and it is suspected that outbreaks may occur when herd immunity, or the proportion of individuals in a population who are resistant to the virus, wanes, and the virus is reintroduced. The most common organs affected by morbillivirus are the lungs, brain, and immune system, resulting in immunosuppression. There are a number of really nasty symptoms associated with clinical disease from the virus, including respiratory and neurologic issues, and secondary infections such as bacterial, fungal, and parasitic infections. Compared to 1987, we now have a number of ways to detect morbillivirus. First, antibodies can be detected in the blood serum, which can indicate exposure to the virus. Second, PCR can be used to detect viral DNA, providing evidence for an infection, which can then be identified by genetic sequencing. Third, immunohistochemistry can be used to detect the virus and provide evidence of infection. And finally, histopathology, or examining the tissues under a microscope, can be used to detect lesions that are compatible with morbillivirus infection and to confirm clinical disease. A combination of these methods are currently used to identify an animal that may be suspect or confirmed positive for morbillivirus. Which leads us to the current unusual mortality event for bottlenose dolphins in the mid-Atlantic. This figure shows the number of stranded dolphins on the y-axis by month on the x-axis. Historical monthly average strandings are in blue with error bars for twice the standard deviation. Beginning in July 2013, strandings began to increase, particularly along the coast of Virginia. Does that sound a little bit familiar? Gross necropsy and histopathology results suggested morbillivirus, and by August, PCR and immunohistochemistry had confirmed an outbreak. August had exceptionally high stranding numbers, with almost 350 strandings, and strandings have remained above average into 2014. The mortalities have followed the same general pattern as the 1987-1988 dolphin die-off. These maps show all bottlenose dolphin strandings by month in green, with morbillivirus confirmed strandings in yellow. And note that we don't test every single dolphin for morbillivirus. So as a state is confirmed to have a positive animal, the outbreak has spread there, we reduce the number of animals that we test. So not all those green dots have been tested. We're really trying to monitor the movement of the virus. So the maps show the morbillivirus off Virginia through New York last summer, and then as the animals moved south into the fall and winter, and they migrated down the coast. The virus itself moved down the coast. Oop, there you go. And our furthest south confirmed stranding right now is from Vero Beach, Florida in December 2013. This figure shows bottlenose dolphin strandings by state and month. And it's a little busy, but darker colors are the northern states and lighter colors are the southern states. And note that the highest mortality occurred here in Virginia at the initial start of the outbreak in August. And mortality increased in the southern states over here into the fall and winter. But the magnitude of the outbreak in the south has been relatively lower than it was during the initial outbreak in the north. 4% of bottlenose dolphins have live stranded during this event, which is higher than historic rates. As I said, bottlenose dolphins don't typically live strand. And many of these live stranded animals are in such poor condition that they expire on their own shortly after stranding. Younger animals, the perinates, which are the animals less than 115 centimeters in total length, and the calves have been more commonly found than adult animals, which would make sense if the virus were impacting animals that had not been exposed during the die-off 25 years ago, so a reduction in herd immunity. And interestingly, males have been found more often than females. In total, 208 animals have been tested for morbillivirus from New York through central Florida, of which 199 are suspect or confirmed positive, or 96%. That's an astonishingly high percentage, indicating a clear cause for the UME. Although we know, because we know that morbillivirus can spread to other cetacean species, we wanted to test, every other animal that's stranded during the event. So we've tested tissues from species, from 13 other species. Of those, 8 were negative, and 5 species were either suspect or confirmed positive. However, there's been no confirmed clinical disease in any non-bottlenose dolphin species except for one striped dolphin. So they have been carrying the morbillivirus, but we don't know that it's actually what's causing them to die, making them sick and die. Although we know the cause of this event, there is still a lot of additional testing and research to be conducted. We need to monitor the spread of the outbreak as the animals return north for the summer and determine when the outbreak itself is over. Per the 1987-88 event, we'll be testing a subset of dolphins to rule out biotoxin involvement. One of the common secondary infections seen in this UME and others is brucella, and so it's a bacteria, and we'll be examining the prevalence of brucella in this event. We will test tissues for persistent organic pollutants, which are known to have a number of negative health and immunosuppressive effects. We'll be working with researchers to model the event and conducting research to better understand the morbillivirus itself, including sequencing the virus and comparing the sequences to other outbreaks, studying how the virus distributes in the body and how it is shed, and conducting retrospective tissue analyses from stranded cetaceans to examine if there might be a reservoir species for the virus. Where is the virus itself living out in the environment? Importantly, we'll also be attempting to determine which of the bottlenose stocks have been affected by this event and by how much, and conducting post-UME monitoring of impacted populations. So we've discussed a UME affecting more than 1,000 dolphins across a broad geographic scale in a short period of time that was clearly caused by a viral outbreak. Next, we're going to switch gears and discuss a highly localized UME in a small portion of the Indian River Lagoon in Florida. The Indian River Lagoon includes 3 water bodies: the Mosquito Lagoon, here in red, the Banana River, here in yellow, and the Indian River, shown in blue. It stretches more than 156 miles from Ponce de Leon Inlet in the north to Jupiter Inlet in the south, and it is a semi-enclosed lagoon that supports a year-round resident population of bottlenose dolphins, the Indian River Lagoon estuarine system stock. It's estimated that the 600 to 800 resident dolphins rarely, if ever, leave the lagoon, and that they show strong site fidelity to specific areas in either the northern or southern parts of the lagoon itself. A recent number of studies have noted significant health issues related to the bottlenose dolphin's distribution pattern within the lagoon, with northern IRL dolphins exhibiting significantly greater health issues than southern IRL dolphins. Prior to 2013, IRL dolphins had experienced 2 recent UMEs in 2001 and 2008. And although the causes of those UMEs were investigated, they were never determined. However, emaciated dolphins were observed in both events. In early spring through late fall 2011, there was a massive bloom of phytoplankton and loss of seagrass throughout most of the IRL system, extending from the southern Mosquito Lagoon to just north of the Fort Pierce Inlet. And the event consisted of 2 concurrent blooms, a lesser bloom down here in the central IRL and a so-called super bloom in the northern part of the system. In addition, the following year, there was a brown tide bloom in the same area, which reappeared in 2013. As a result of the 2011 phytoplankton blooms, coverage of seagrass beds in the lagoon was reduced by approximately 60%, or 47,000 acres. So enter the 2013 bottlenose dolphin UME. In January 2013, elevated bottlenose dolphin strandings began to be noticed in the northern and central Indian River Lagoon. These strandings coincided with elevated pelican and manatee mortalities in the same area. As of February 2014, there are a total of 82 stranded dolphins in this event, of which a majority are older adults, with a few juveniles. So how does that compare to the historical average? This figure shows stranded dolphins on the y-axis and month on the x-axis. Historical averages with 2 times the standard deviation error bars are in blue. 2013 strandings are in red, and the 2008 unusual mortality event is in green. 2008 non-UME months are noted in yellow. You can see that the 2013 UME was longer in duration and higher in magnitude than the 2008 UME. And the UME appears to have ended in September 2013, with strandings returning to at or below average. To investigate the causes of this UME, our Tier 1 investigation focused on histopathology. Preliminary findings have found emaciation in 83% of the stranded dolphins, but otherwise there are no consistent pathologies or causes of death. As part of our Tier 1 investigation, Hub's SeaWorld Research Institute, our stranding network partner in the area, also conducted surveys of the IRL to evaluate body condition and document any unusual behaviors in free-swimming dolphins. As compared to 2012, where surveys noted only 1% of dolphins were underweight, in August